Once again, my dear viewers, welcome to Future Knowledge Bank. I am very thankful to you as your response is very good in the respiration part 1 in biology, and now I am giving the respiration part 2 and completing the plant wise. In the part 1, I given the human respiratory system point of view and some points of the human respiratory system also I am going to say now. Apart from that one, and total anaerobic, aerobic respiration and that what kind of the different activities are there within this respiration I am giving in front of you now. First of all, now initially I want to say about what is aerobic respiration and what is anaerobic respiration. The thing is mainly aerobic respiration is nothing but the presence of oxygen. The presence of oxygen, whatever the respiration is taking place, that is called aerobic respiration. Respiration in the sense people will think that one exchange of gases, that is completely wrong. Once again I am saying that is not even wrong. Just whenever the gas oxygen is taking in, carbon dioxide releasing out, during this process lot of energy molecule break down and so much energy is going to be produced and developed. With that energy the human can live. So respiration is main motto is releasing of energy, not just breathing. So here aerobic respiration in the sense is presence of oxygen and anaerobic respiration is absence of the oxygen. And what are the things that is going to be comes out from the aerobic respiration is carbon dioxide, water and energy. And comes to the point of anaerobic respiration, here ethanol and lactic acid and carbon dioxide is going to be released. And aerobic respiration takes place in cytoplasm and mitochondria. And anaerobic respiration takes place only in cytoplasm of the cell. And that means once again I will repeat you, aerobic respiration takes place both in cytoplasm as well as mitochondria. Anaerobic respiration takes place only in the cytoplasm. And here we human being is getting total net of 40 ATP molecule. That means adenosine triphosphate molecule, which is giving enormous energy for the living of the human. In that 38 ATP molecule just coming from aerobic respiration. So aerobic respiration is very must for us. In that 38 ATP molecule from aerobic respiration, only 2 ATP molecule from anaerobic respiration. And now the aerobic respiration takes place in especially in all multicellular plants and animals. And anaerobic respiration takes place in bacteria, fungi and other organisms. So these are very key points and key differences that you are going to see between aerobic respiration and anaerobic respiration. Now, one more point that you are going to see here on the chart, I will explain you later before giving one more point in front of you. What it means, in our body, one part is there between stomach and stomach and lung, buccal cavity, and that is called diaphragm. This diaphragm is absent. If the diaphragm is absent in the human beings, what kind of problems the human is going to face? The main question is, if the diaphragm is not present in the human, what is the problem is going to face? Means, here you see two types of major problems you are going to face. First one is congenital problems. That means automatically whenever the person who is not having the diaphragm, in the couple not having the diaphragm, the children will going to take birth with a lot of defects by birth only, not having the limbs or eye defects or heart is something wrinkled, irrespective of that one, and we cannot even say what will be the reason also. So this is very dangerous if the diaphragm is absent. Congenital defects. That means congenital defects means in the sense is the defects which we are getting by birth itself. Now comes the point of another one, acquired defects. Acquired defects means the defects little bit accident also, the problem will become severe. If that little bit accident also, the problem will be severe. That is going to call acquired defects. So diaphragm is not there, congenital defects and acquired defects we are going to face. Now I am ready to explain the chart one here. Interestingly, you can see in this chart what we are going to do and how the carbon dioxide is producing by breaking down of glucose here. And this experiment, first of all, one test tube I am taking here. You can see here in this test tube I am first of all providing the glucose here. Whenever I am pouring the glucose, in this glucose lot of oxygen is there. Ce6H12O6, that you know very well, lot of oxygen is present in this glucose, and I want to remove the total glucose content in this one. So what to do? I want to boil this glucose. I want to boil this glucose and let the glucose is going to be cooled down. Let the glucose is going to be cooled down. During this process, and lot of oxygen is going to be released from this glucose. And okay, the oxygen is released. And here, how can we identify the oxygen is emitted from the glucose here? What is a test? What is a test? We have to go for the glucose released. In this case, means we are going to apply Janus green B solution or diazine solution. Janus green B solution or diazine solution. If we pour this solution in the glucose, already the glucose was boiled and cooled it down. And you can see the pink color is going to be formed. The pink color is going to be formed means that oxygen is already left the glucose solution. No, there is no oxygen in this glucose solution, that you understood. If oxygen is not there, what will be the other solution? Carbon dioxide will be there. There only two gases will in the living organisms. One is either oxygen should be there or carbon dioxide should be there. And there won't be any vacuum over here. So now we confirm that one. After boiling the glucose, cooling down, lot of oxygen is emitted out, and we want to test whether the oxygen is emitted out or not. We are going to pour diazine solution or Janus green bean solution, that is going to give the pink color if oxygen is released. If it won't give pink color if oxygen is not released. So now pink color we got, then there is a chance here in this from this delivery tube, there is a chance oxygen can enter. So our experiment is we should not make oxygen entry here. We don't want oxygen entry here. So what kind of step we have to take here? We have to put certain paraffin liquid here. You can see where I am showing the pointer lighter. You can see paraffin layer. One paraffin layer providing. This paraffin layer is not allowing the outside oxygen to enter into the oxygen less glucose. Oxygen less glucose. So there is no oxygen inside the glucose. There is no oxygen moving outside that one. So only in this test tube, what is providing total arena? Carbon dioxide only is forming in this glucose. This carbon dioxide will comes out through the delivery tube like this. It will come, carbon dioxide is going to come through the delivery tube. And what is the evidence that gas is so evolved is carbon dioxide? And carbon dioxide gas vachindi ani confirmation etla ante in this second test tube already am keeping the lime water. I am already keeping the lime water, that is a slaked lime. This colorless lime water, if the carbon dioxide is received, this colorless lime water turns milky white. This is turns milky white. If it is turned milky white, you can confirm carbon dioxide is produced in this test tube from the glucose and passed through the delivery tube and reached the test tube. So carbon dioxide only can form the milky white color in this one. So this is nothing but how to how to see that one anaerobic respiration developing the carbon dioxide from this experiment. And a 1 minute break, I will come with another experiment in front of you. Before going to explain this experiment here, germinating seeds experiment in the thermos flask, I am giving one important point regarding the respiration. See, whenever we are playing the games, the children is playing the games, or elder people are worked so much, మీ ఇంట్లో చాలా పని చేస్తే పెద్దవాళ్ళు అందరూ ఎక్కువ పని చేసినప్పుడు చాలా అలసిపోయాను, నాకు చాలా బాడీలో పెయిన్స్ వచ్చేస్తున్నాయి, ఎందుకు పెయిన్స్ వస్తున్నాయి, పిల్లలందరూ అలసిపోయారేంటి, ఇటువంటి డైలాగ్స్ మీకు రావచ్చు. అండ్ దిస్ ఇస్ ఐఎమ్ గివింగ్ ఇన్ తెలుగు బికాస్ ఇది చాలా ఇంపార్టెంట్ పాయింట్ కాబట్టి నేను తెలుగులో వేస్తున్నా. అంటే ఎప్పుడైనా బాగా ఎక్సర్సైజ్ చేసినప్పుడు కానీ ఆడినప్పుడు కానీ పెద్దవాళ్ళు బాగా పని చేసినప్పుడు వాళ్ళకి ఒళ్ళు నొప్పులు కాళ్ళు నొప్పులు ఇవన్నీ వచ్చేస్తాయి. కారణం ఏంటంటే అక్కడ మెయిన్గా గ్లూకోజ్ లాక్టిక్ యాసిడ్ కింద కన్వర్ట్ అయిపోతుంది. ఇన్ ద బాడీ, ఇన్ ద సెల్స్, ద గ్లూకోజ్, వాట్ ఎవర్ ద గ్లూకోజ్ ఈస్ ప్రొడ్యూస్డ్, దట్ ఈస్ గోయింగ్ టు బి కన్వర్ట్ ఇన్ టు లాక్టిక్ యాసిడ్. లాక్టిక్ యాసిడ్ సెల్స్ మజిల్స్లో తయారైందంటే ఆటోమేటిక్గా బాడీ పెయిన్స్, హ్యాండ్ పెయిన్స్, అన్ని పెయిన్స్ వచ్చేస్తాయి. ద స్ట్రెయిన్ అయిపోయి గ్లూకోజ్ అంతా కరిగిపోయి లాక్టిక్ యాసిడ్గా ఫామ్ అవుతుంది. లాక్టిక్ యాసిడ్ ఈస్ ద మెయిన్ థింగ్ టు ప్రొడ్యూస్ ద మజిల్ పెయిన్. సో దిస్ ఈస్ ద interesting situation. Accumulation of the lactic acid in the muscle cells is going to be considered as the pain in the other people. Okay. Now another one is What is the cellular respiration? What is the cellular respiration? When in the cells, the glucose molecules are break down and lot of energy is going to be produced. Glucose is break down, lot of energy is produced within the cells, and that is called cellular respiration. This takes place in the prokaryotic cells. So now directly coming back to this experiment, here I am taking two thermos flask here, two thermos flask. One thermos flask I am keeping the one dried seeds. In one thermos flask I am keeping the dried seeds, one cork I am keeping, and one thermometer I am keeping to measure the temperature. And in another, this one is a thermos flask only. Here I kept germinated seeds. Here I kept germinated seeds and మొలకలు పెట్టాను ఇక్కడ, మొలకెత్తిన శనగలు పెట్టాను ఇక్కడ. థర్మస్ ఫ్లాస్క్ అరేంజ్ చేశాను, థర్మామీటర్ కూడా పెట్టాను. టెంపరేచర్ ఏమైనా పెరుగుతుందా లేదా? ఈ ఎక్స్పెరిమెంట్, బై దిస్ ఎక్స్పెరిమెంట్ వాట్ ఈస్ మై aim means to show that one lot of heat is developed during the from the germinating seeds. Germinating seeds నుంచి ఎంతో హీట్ వస్తుంది అని చెప్పడానికి కోసము. So now dry seeds తీసుకున్నాను. I took the dry seeds, I kept in one thermos flask and fixed the cork and arranged thermometer. I am keeping 3 to 4 hours continuously. That from the dry seeds, the thermometer present in the flask, no rise in temperature. There is no change in temperature. That means dried seeds are unable to develop any sort of heat. And here, but in this case, you can see, in this case, germinating seeds I kept and thermometer I kept. Suppose thermometer reading is 10 degrees temperature here, and after certain occasion, this I provided germinating seeds here, germinated seeds, and temperature is went to 25. That means 15 degrees temperature is raised. Here, this kind of raise, that means during the production of the seed into plant, plumule and radicle, we call the parts of the seed plants are small, tiny plants, parts we call plumule and radicle. These plumule and radicle, while they are taking birth from the seed, lot of heat is going to be released. Due to the release of heat, the thermometer shows higher temperature. From this experiment, what you understood, the germinating seeds are providing lot of temperature. Okay, and in this way, the respiration, most important topics I already covered, but in front of the SA1 exams, totally important objective and important questionnaire. Once again, I will deal in completely, not in part 1, part 2 type, completely one summative, concise matter I will give in this bio and physics and maths also. Okay, thank you for watching.